An animal cell can be seen under a light microscope and can also be seen under an electron microscope. Light microscope में मैंने आपको बताया था कि a light microscope has different powers और objective lenses of different powers. इसमें ये एक rounded body है which is known as nose piece, and it has one or two eyepieces attached with it here. ये eyepieces हैं. And this is nose piece. There are 3 or 4 objective lenses attached here. हमने पहले भी डिस्कस किया था. And each objective lens has some power written on it. और ये जो powers इसके ऊपर लिखी होती हैं, they may be X4, X10, X40 and X100. And there may be power written on eyepiece as well, which may be an X10, which may be an X20, which may be an X8, X7 or any power. So eyepieces may have different powers. और इसी तरह से ये जो ऑब्जेक्टिव लेंसेज हैं, ऑब्जेक्टिव lenses have different powers. Okay, eyepieces की भी different power होंगी, and we will also have powers of different powers of objective lenses as well. इनमें से कोई एक ऑब्जेक्टिव लेंस हम यूज करेंगे at one time, और जो हम यूज करेंगे that will determine the power of this microscope. जैसे फॉर एग्जांपल अगर हम यहाँ से ये 4 लेते हैं और आईपीस हमारा 20 टाइम्स मैग्निफिकेशन है, तो दिस विल बी x4 into x20 and this will give us x80. इसी तरह से अगर यहाँ से हम 100 लें और यहाँ से हम 20 लें, तो दिस विल बी x100 into x20, this will make x2000. So this is the maximum power or maximum magnification of a light microscope. Got that? ये lowest power है. This is lowest power. And this is highest power. Both these powers are not in our syllabus. Okay? These are not in our syllabus.
You have written 200 instead of 20.
Sorry?
Written into 200 instead of 20.
Okay. Yes. Thank you. अच्छा, अब इसमें से ये वाली जो दो हैं, ये हमारे सिलेबस में है. इसमें से X10 को हम कहते हैं लो पावर और X40 को हम कहते हैं हाई पावर. Okay. However, you will see the animal cells diagrams that have been seen at maximum power of a light microscope at 2000 times. तो सबसे पहले हम देखते हैं कि animal cell seen under a low power of a लाइट माइक्रोस्कोप। अगर हम इसे लो पावर के नीचे देखें तो ये हमें एक आउटर बाउंड्री नजर आएगी, मे बी द चीक सेल्स और सम अदर सेल्स, एंड वी विल सी अ डार्कर central region. That's it. This space is filled by a fluid called cytoplasm and a centrally densely stained body is known as nucleus. So we will only label these two structures under a light microscope. We will never label cell membrane. Cell membrane is not to be labeled. Why? Because it is not seen under light microscope. तो जो ऑब्जेक्ट हमें नजर नहीं आ रहा, हम उसको लेबल भी नहीं करेंगे लाइट माइक्रोस्कोप के नीचे. तो वी विल ओनली लेबल साइटोप्लाज्म एंड न्यूक्लियस, दो वी नो दैट दिस इज एन आउटर बाउंड्री which will have cell membrane. You got a point? If you look at the same animal cell, seen under a light microscope, at high power. यह हाई पावर का मतलब क्या है? It is x 2000 times, यानी highest power, which is not achievable in the school microscope. इसलिए ये सारे स्ट्रक्चर्स हमें स्कूल माइक्रोस्कोप में नहीं नजर आएंगे. ठीक है, बुक में हमें नजर आएंगी डिटेल्स इसकी, but we will not be able to see it under the microscopes in our school. इसलिए कि जो स्कूल माइक्रोस्कोप्स होती हैं, they are academic microscopes. इनकी जो मैग्निफिकेशन है, that is not what has been written on them. There is another kind of microscope which is known as analytical microscope. जो एनालिटिकल माइक्रोस्कोप्स होती हैं, उसके ऊपर जो माइक्रोस्कोप की पावर लिखी होगी वही पावर एक्चुअली में होगी. Okay, so you will not be able to see all these details under your school microscopes, but however, you will be able to see a larger cell. We will not label cell membrane, as cell membrane is not seen. We will see nucleus in the center. We will also see two more structures in the nucleus, and maybe we can see chromatin as well. So this whole structure is nucleus. These are nucleoli, or singular will be nucleolus, and this will be chromatin. Obviously this is cytoplasm here. Within the cytoplasm, we will see mitochondria. And other than mitochondria, we will be able to see Golgi bodies and centriole. There will be some structures that are unidentifiable. Unidentifiable. These are the structures that we will be able to see under a high power of light microscope. look at an animal cell seen under an electron microscope. So obviously an outline of the animal cell will be the same, but इसमें हमें जो nucleus नजर आएगा, nucleus will have two membranes इस तरह से. Okay, so this is inner membrane of the nucleus. We will also see an outer membrane. So this is an outer membrane of the nucleus. It is joined or fused with the inner membrane at points where it makes nuclear pores. So these are nuclear pore. Okay, and this is outer membrane, outer nuclear membrane, and this is inner nuclear membrane. Collectively, both these are known as nuclear envelope. इलेक्ट्रॉन माइक्रोस्कोप के नीचे जब हम इस न्यूक्लियर मेम्ब्रेन्स को लेबल करेंगे तो वी विल लेबल देम एज न्यूक्लियर एनवेलप rather than nuclear membranes. Okay, then we will see two more structures inside it known as nucleolus and chromatin. So this is how nucleus will be visible under an electron microscope. The outer membrane, this is an outer membrane. It is extended outwards, making a network of tubules which extends to the cell membrane. Okay, so this network of interconnected tubules or channels is known as endoplasmic reticulum. This is called endoplasmic reticulum. Okay, there are two types of endoplasmic reticulum. One that is towards the nucleus has ribosomes attached on its surface. इसके यहाँ पे ribosomes होंगे. And this is known as rough endoplasmic reticulum. This stands for rough endoplasmic reticulum. Okay. Further on the outer side, it has another type of endoplasmic reticulum which doesn't have any ribosomes attached on it, and it is known as smooth endoplasmic smooth endoplasmic reticulum. Okay. Within the cytoplasm, closer to the nucleus, we will also see flattened membrane-bound sacs which is known as Golgi body or Golgi apparatus or Golgi complex. So we will write Golgi bodies here. From the surface of Golgi bodies, we will see small membrane-bound vesicles will be pinched off, and these are known as Golgi vesicles. इनको हम कहेंगे Golgi vesicles. Other than these Golgi vesicles, we will also find double membrane bound cell organelles called mitochondria. And within the cytoplasm, we will see ribosomes. And near this nucleus, we will see a star-shaped structure consisting of two cylinders known as centrioles. Whole this structure is enclosed in a cell membrane on its outer side, which is known as cell membrane. Okay, so this is how an animal cell will look like under an electron microscope. We will also have small membrane-bound vesicles that may be known as lysosomes. Or other than that, we will have cytoskeleton, and these are microvilli. So this is the detailed structure of an animal cell seen under an electron microscope. We will discuss these components one by one.